All right. Hello, everyone, and welcome to today's webinar, Automated GMP-compliant PSC Banking in a Closed System. I'm Isabel, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I would like to now welcome our speakers, Rafal Krol, Principal Investigator in the Research and Development Center at CiRA Foundation, and Chao Sheng, Product Manager for Regenerative Medicine at Miltenyi Biotec. Chao, you may now begin your presentation.
Okay, thanks a lot, Isabel, for the nice introduction, and welcome everyone to our webinar also from my side. My name is Chao Sheng, and I am the Global Product Manager for the Regenerative Medicine portfolio at Miltenyi Biotec. Today, we are very glad to have Dr. Rafal Krol from CiRA Foundation with us, and he will present some of his recent progress on banking of GMP-compliant human pluripotent stem cells. I know that his projects are still ongoing, so we are very happy to have this opportunity today to already see some of his most recent data. And we are also very happy that our technologies, especially the CliniMACS Prodigy cell manufacturing platform, can support his projects on PSC banking. Well, before Dr. Krol's presentation, I will first very quickly introduce you our solutions for automated PSC expansion and differentiation, as well as standard flow cytometric QC analysis with our widely used CliniMACS Prodigy cell manufacturing platform and the MACSQuant flow cytometric analyzer. Well, looking at the general steps of PSC applications, in particular, regenerative medicine, there are many important requirements during the manufacturing process. For example, it needs to be GMP compliant, scalable, and reproducible. Meanwhile, to make the final cell products affordable to the patients and also to the healthcare system, the manufacturing process also needs to be very cost-effective. So for all these reasons, manufacturing PSCs and their derivatives for clinical applications is such a challenging and complex endeavor. And most of the current manufacturing processes involve multiple, very expensive instruments, very skilled, very experienced operators, and high-class clean rooms. And many open and separated handling steps are also introduced into these workflows. Meanwhile, standard in-process control and quality control analysis are required throughout the whole manufacturing process. In addition, in some applications, the desired cell populations also needs to be isolated in a sterile, gentle, and GMP-compliant environment. So all these factors together could really limit the manufacturing skill, compromise the reproducibility, at the same time, drive up the manufacturing cost. So how to make these very complex cell manufacturing processes more standardized, more scalable, at the same time, more affordable for the end users? Well, here are the solutions from Miltenyi Biotec. So with the CliniMACS Prodigy cell manufacturing platform, these separated open handling steps, these multiple the instruments, devices, the skilled operators, and high-class clean rooms, they can be integrated into one automated enclosed manufacturing system. And the users can also build customized PSC expansion and differentiation processes on the CliniMACS Prodigy. With the MACSQuant analyzer, standard in-process control and quality control assays can also be realized by automated flow cytometry analysis. Finally, the MACSQuant title cell sorter can be applied to isolate the desired cell populations with a very gentle, fast, and GMP-compliant cell sorting technology. So now let's just very quickly have a look at the hardware features of the CliniMACS Prodigy. Well, together with the GMP-compliant single-use tubing sets, the CliniMACS Prodigy allows automated cell processing from starting materials to the final products. And upon your request, it can automate all these different cell handling steps, including sample preparation, dense gradient centrifugation, cell washing, medium change, etc. They can also all be automated on the CliniMACS Prodigy. And in the adherent cell culture process, the cell cultivation can actually take place in two different units, the centric unit or in the external culture vessels. First, in the centric unit, so there's a CliniMACS Prodigy chamber lies inside, and it supports automated adherent and non-adherent cell cultures. So you could automate your cell feeding, cell harvesting, you can control the temperature and also the concentrations of air and CO2. We also have a camera integrated under the chamber so that you could visualize your cell cultivation. And there are also multiple input ports are available for the cell cultures. So this CliniMACS Prodigy chamber offers 100 square centimeter surface space. Well, if larger surface space is required, then the external culture vessels can also be very easily connected to the CliniMACS Prodigy tubing sets via serial welding. For example, these multi-layer cell stacks. So the users can actually connect maximally 3 of these 5-layer cell stacks at the same time. And with 3 5-layer cell stacks, then you could have roughly 10,000 square centimeter surface space for the cell culture. Well, now I would like to shift your attention to the adherent cell culture software. So when we designed this software, our goal is to make sure that the software is flexible enough to culture different cell types. So that is why we modularize the individual cell handling steps. So now you can see these different modules lies on the main menu of the adherent cell culture software. So for example, we have the dense gradient centrifugation module, the coating module, inoculation, culture, medium change, and the harvest modules. And the users can actually freely select and combine these different modules. As a result, very complex workflows, for example, directed differentiation of human pluripotent stem cells can be realized on the CliniMACS Prodigy adherent cell culture process very precisely. And within each module, the users can also set up the cultivation parameters according to their established SOPs. For example, in the coating module, the users can set up the coating temperature, the coating time, the volume of the coating solutions, all according to their established SOPs. So these are the CliniMACS Prodigy features. Now, also very quickly, I want to introduce you our standard flow cytometry QC analysis. So for cell manufacturing process, we know that requires stringent QC analysis. And the MACSQuant analyzer is equipped to meet the needs in regulated cell manufacturing. And this instrument offers a lot of automated operational features, which could make your work not only comfortable, but also it can streamline and standardize the workflows. So from startup to shutdown, this includes washing steps. The MACSQuant analyzer takes on labeling and the processing of the samples and standardize the initialization with automated calibration or compensation. The accurate volumetric cell count can also be performed on the sample itself and does not require any addition of further beads. So to automate also the acquisition and analysis of the IPC QC steps, we also offer several on-demand express mode packages. So these are algorithm-based gating tools that can be added to the MACSQuantify software and allows for unbiased and user-independent analysis, resulting in reproducible flow cytometry data. In addition, if you need an express mode for your specific QC application, then we can also customize the express mode to your data analysis. And with the Smart Gain technology, you can perform multiple instrument alignments for consistent and reliable measurements from instruments to instrument, no matter of the operator or where the instrument is located. In addition, the software can also help you with documentation. So the MACSQuantify 21 CFR Part 11 software delivers everything from audit trial to a good user and role management system, which is necessary to validate the system compliantly. So now I would just very quickly also show you some of our results of PSC expansion and differentiation using the CliniMACS Prodigy and the MACSQuant analyzer. So we have established several workflows. For example, the GMP-compliant expansion of human mesenchymal stromal cells and human pluripotent stem cells, and also differentiation of human PSCs into midbrain dopaminergic progenitor cells. At the moment, we're also working on a new workflow to differentiate human pluripotent stem cells into cardiomyocytes on the CliniMACS Prodigy. And today, we will focus on these two workflows in the middle. So for PSC expansion, we have developed the GMP-compliant iPSC Brew GMP medium. In using this medium on the CliniMACS Prodigy, the users can choose and combine already different modules to automate all the complex handling steps. For example, coating of the surface, PSC inoculation, medium change, PSC splitting, and harvesting, etc. on the CliniMACS Prodigy. So we also performed side-by-side comparisons between menu and CliniMACS Prodigy-based PSC expansion using the reaffinity recombinant antibodies and the STAMX tri-linked differentiation kit. And looking at the expression levels of these key pluripotency markers and the tri-lineage differentiation potentials, we can see these two groups, the CliniMACS Prodigy versus manual control. These two groups really gave rise to very comparable results. And then by using the centric unit and different sizes of the external culture vessels, the users can also easily reach different PSC expansion skills on the CliniMACS Prodigy. We know that establishing GMP-compliant PSC banks is a major step in many clinical applications. And for this purpose, the closed and automated CliniMACS Prodigy adherent cell culture process, together with the iPSC Brew GMP medium and our standard QC reagents, these could be really valuable assets for your PSC banking projects. Not only the expansion, the CliniMACS Prodigy adherent cell culture process can also realize very complex PSC differentiation processes. For example, differentiation into midbrain dopaminergic progenitor cells. So we have collaborated with Dr. Marlene Palmer and Dr. Agnete Kirkby within the EU Neuro Stem Cell Repair Consortium and have also already very successfully adapted their very efficient manual differentiation processes into our closed and automated CliniMACS Prodigy. So this is the process overview. And here I also want to show you some side-by-side comparison between manual and the CliniMACS Prodigy. Using the MACSQuant analyzer again, so the results show that the cells differentiated on the CliniMACS Prodigy are very comparable to the cells that differentiated manually, with more than 80% of the resulting cells co-expressing the key MDA progenitor markers FOXC2 and OTX2, and nearly no expression of the unspecific or pluripotency markers. In addition to the good differentiation efficiency, the CliniMACS Prodigy also offers automation as well as closed and GMP-compliant environment. Meanwhile, it is also scalable. So in this case, we started with 1 million iPSCs, and then after 5 days pre-expansion and another 16 days differentiation, we could harvest roughly 3 to 4 billion MDA progenitors, which can be translated to roughly 600 to 800 potential patient doses, considering 5 million cells per unit. And the users can also further scale it up or scale it down according to their technical request. So for the end users of the CliniMACS Prodigy adherent cell culture process, we also offer a very comprehensive training program. So this is a glance of this training schedule. So it is a 3-day training program, including theoretical sessions, guided and unguided hands-on sessions, and troubleshooting sessions. So it is very helpful for the users to get familiarized with the CliniMACS Prodigy. In fact, Dr. Krol has also participated in this training in the past. Yes, the CliniMACS Prodigy adherent cell culture process is still quite new, but there are already quite a number of labs worldwide started to automate their PSC or MSC workflows on it. For example, Professor Annelise Benesser-Guseli's lab from INSERM in France. So in her presentation at IST 2020, she demonstrates that the CliniMACS Prodigy provides a safe, standardized, and scalable manufacturing platform for GMP iPSC master cell banks. And this presentation is freely available on our website. So today, Dr. Krol will continue the discussion on this very important topic. This is the disclaimer of my presentation. Okay, so that's actually all from my side. So if you want to discuss any questions in a separate meeting, so you're welcome to contact me via email. Thanks a lot for your attention. Now I would like to give the stage to Dr. Krol to talk about his recent work on GMP-compliant PSC banking at CiRA Foundation.
Thank you very much, Chao, and welcome everyone. My name is Rafal Krol, and I am a principal investigator at CiRA Foundation's Research and Development Center. My presentation will consist of two parts. Firstly, I will introduce CiRA Foundation and talk a little about its structure and goals. Then I am going to summarize our progress in process development for transition from manual small-scale maintenance of iPS cells onto automated GMP-compliant closed culture system. CiRA Foundation, an offshoot of Kyoto University's Center for iPS Cell Research and Application, manages iPS cells manufacturing and quality control and acts as a bridge for the transfer of iPS technology to industry. It was founded in September 2019 and has been registered as a public interest incorporated foundation on April 1st, 2020. After more than 10 years since the first human iPS cells were reported, iPS cell technology is being used to create new medical therapies. CiRA is involved in many projects aimed at testing the safety and efficacy of those therapies. The facility for iPS cell therapy, the basis of the CiRA Foundation, has been responsible for managing the iPS cell stock project, manufacturing and quality control of iPS cells for clinical applications. The iPS cell stock project has been financially supported by the Japanese government and has received tremendous assistance from the Japanese Red Cross Society and the Japan Marrow Donor Program. As of April 2020, it consists of 27 iPS cell lines from 7 donors. The cells have been used in clinical trials managed by RIKEN, Osaka University, and CiRA. We provide services for academic and industrial customers in cell manufacturing, quality assurance and storage, research and development, clinical application support, training and human resources development, and information sharing in industry, government, academia network at the national and international levels. The core of our philosophy is to provide the best iPS cell technology with affordable price. In order to deliver medical care using iPS cells to the patients as quickly as possible, we act as a go-between to bring technologies such as cell manufacturing and quality assessment to the industrial world. Our approach to ever-evolving technology is to strive every day for continuous improvement. By integrating and sharing research and development information, we contribute to clinical applications, improved quality and safety, and cost reduction of regenerative medicine. We continue to drive our public service through strict adherence to ethical standards. Here is a short timeline of CiRA Foundation's history. After discovery of iPS cells by Professor Yamanaka, Center for iPS Cell Research and Development was established at Kyoto University, and Cell Processing Center facility for iPS cell therapy was completed in April 2010. In August 2015, iPS cell stock project was launched. In February 2017, second Cell Processing Center, FITS2, was completed at CiRA. In April 2020, CiRA Foundation has been registered as a public interest incorporated foundation. The central part of CiRA Foundation is Facility for iPS Cell Therapy, consisting of quality and manufacturing sections. I am affiliated with another unit, Research and Development Center. Finally, there are two support units, Planning and Administrative Departments. The representative director of CiRA Foundation is Professor Shinya Yamanaka and executive director, Professor Naoko Takasu. The term valley of death is often used to describe the situation where technologies and ideas developed at universities and other academic institutions do not successfully transfer to the industry. Universities and research institutions usually do not have the effective tools to put their ideas and discoveries to practical use. In the US, a bridge over the valley of death is often made by venture companies or large investments, but in Japan, this kind of infrastructure is rare. The CiRA Foundation is building a bridge for regenerative medicine by providing iPS cells, which require great cost and advanced technology to manufacture, free of charge to non-profit institutions and at a low price for profit institutions. Now I would like to shortly describe our ongoing projects. iPS Cell Stock Project is collecting blood and generating iPS cells from HLA homozygous donors because there is increased chance of positive donor-recipient matching such cells. The stocked iPS cells undergo strict quality control and are distributed upon request to hospitals and research centers for clinical applications. We estimate that the current iPS cell stock at FIT could match 40% of the Japanese population. Currently, we have 27 lines prepared from 7 donors, and some of these lines have already been used in iPS cell-based clinical cell trials. To be able to provide therapies for remaining 60% of Japanese population and also world population, we started to generate HLA genome-edited iPS cell lines. They are designed to reduce the risk of immune rejection by knocking out HLA A, B, CT2 and retaining HLA C and E in order to escape T and NK cell-mediated immunity. We estimated that only 7 HLA genome-edited iPS cell lines could cover 95% of the Japanese population, and 12 cell lines could cover the entire world. My iPS is a project aimed at providing any individual with their own iPS cells at a reasonable price. To make such therapies available for large population, CiRA Foundation is developing technologies that will allow manufacturing of one's own iPS cells at a cost of 1 million yen, which is roughly $10,000 by 2025. CiRA Foundation and its core Facility for iPS Cell Therapy is responsible for manufacturing and banking of clinical-grade induced pluripotent stem cells. It requires efficient and scalable cell expansion protocols that allow for maintenance of cell morphology, immunophenotype, genetic stability, and pluripotency under current good manufacturing practice requirements. We are currently working on the transition from existing manual cell expansion protocols in the iMatrix 511 StemFit AK03N culture system onto an automated closed culture platform, CliniMACS Prodigy Adherent cell culture system that allows GMP-compliant and scalable iPSC cultivation. Here we report the progress of the process development. The Preliminary requirement for the process was that cells from existing stock are to be initially expanded on the standard 6-well tissue culture plate due to the low cell count in our existing stock and to assure good cell quality. Then harvested cells were to be inoculated into the CliniMACS Prodigy TS730 tubing set chamber, cultured under the Adherent cell culture process, then harvested and inoculated once more into the CellStack 1 or CellStack 5 chamber from Corning before final harvest and banking. We hope that the process will allow for the semi-automated expansion of iPSCs from the 100,000 up to 500 million cells scale within about 2 weeks. Since the CellStack chambers are available in several sizes, that process could be highly scalable, and utilization of automated system could greatly reduce the workload of large-scale expansion of iPS cells. Here's the summary of the plan based on the Miltenyi Biotec protocol. 100,000 cells from the stock will be plated in one well of 6-well plate, then expanded cells harvested and inoculated into the Prodigy chamber, passaged once into CellStack 1 or 5, and finally harvested for banking. We hope to produce at least 60 million cells from CellStack 1 and at least 300 million cells from CellStack 5 within 12 to 15 days. From the next slide, I will discuss results of actual experiments. Our standard iPS culture system consists of the iMatrix 511 substrate at a concentration of 0.25 microgram per square centimeter, applied to the vessel without pre-coating, and stem-feed AK03N medium from Ajinomoto. We usually plate 100,000 cells in one well of 6-well plate and are able to harvest around 2 million cells on day 5, dissociating cells with EDTA. The microphotograph depicts usual cell morphology before harvest. Since Chao already introduced the CliniMACS Prodigy instrument, Adherent cell culture software, and workflow for PSC expansion in his presentation, I will concentrate on the protocol and omit handling of the instrument. For the passage into the Prodigy system, we pre-coat the 100 square centimeter chamber with 25 milliliter of 4.5 microgram per milliliter of iMatrix 511, aiming at the chamber coating concentration of 0.5 microgram per square centimeter. This part had to be optimized since coating efficiency of the Prodigy chamber differs from the standard culture vessel, and we needed to account for some protein loss due to the deposition on the tubing. After inoculation of 2 million cells, on day 5 we can harvest around 12 million cells and proceed with the inoculation of external vessel. The microphotograph shows representative image of the Prodigy chamber on day 2. Depending on desired scale of the expansion, we choose appropriate cell stack. When passaging cells into CellStack 1, the 636 centimeter vessel, we use 100 milliliter of 3.5 microgram per milliliter of iMatrix 511 without pre-coating, aiming at the coating concentration of 0.25 microgram per square centimeter. The yield on day 5 after inoculation of 2 million cells is usually around 12 million cells. Again, the microphotograph shows representative image of the culture on day 4. When CellStack 5, the 3,180 square centimeter vessel, is selected for passage, we use 500 milliliter of 4 microgram per milliliter of iMatrix 511 without pre-coating, aiming at the coating concentration of 25 microgram per square centimeter. Here we are still optimizing harvest protocol. Currently, we are able to harvest around 400 million cells on day 5 after inoculation of 22 million cells. But to be able to recover all cells from CellStack 5, harvest timing and confluency need to be perfect. We believe that with optimal harvest protocol, we should be able to get at least 600 million cells. The microphotograph shows a representative image of the culture on day 4. The poor quality of the image results from the fact that to place the cell stack 5 on the standard culture microscope stage, we need to remove the condenser unit, which results in suboptimal illumination and lack of phase contrast. Because we are still working on the process, we did not yet perform extensive quality assessment of the cells expanded in CliniMACS Prodigy system. However, when we return the cells to the standard maintenance system, they seem to retain proper cell and colonies morphology. Full QC is scheduled for after completion of the process protocol. I will finish my talk by short summary. CliniMACS Prodigy Adherent Cell Culture Platform allows for highly scalable iPS expansion in iMatrix 511 StemFit AK03N culture system. Transition from manual PSC expansion system to CliniMACS Prodigy has its challenges, but they can be mitigated without much difficulty. We are working on finalizing the process and planning thorough quality assessment of the cells expanded with CliniMACS Prodigy platform. Thank you very much for your attention. I think that now we will proceed to the QA session.
Right. Thanks a lot, Rafal and Chao, for your informative presentation. So we will now start the live Q&A portion of the webinar. So if you have questions you'd like to ask, please do so now. Just click on the Ask Question box located on the far left of your screen, and we will answer as many questions as we have time for. Right, so let's get started. I can already see the first question, and this is, how difficult is it to adapt manual PSC expansion process into the automated CliniMACS Prodigy system?
Okay, so well, there are two main requirements for successful transition from manual PSC expansion system to CliniMACS Prodigy platform: culture protocol conversion and handling of the instrument itself. We have experienced some challenges when we started to use the CliniMACS Prodigy system, since it's a technology which is very different for manual cell culture, but it does not take a long time to get familiar and comfortable with the system. The big help was the CliniMACS Prodigy Adherent Cell Culture System user training, an intensive 3-day program held at Miltenyi University in Bergisch Gladbach, Germany, that I have participated in before commencing experiments. With regards to the culture protocol optimization, besides becoming comfortable with tubing, bags, and other closed culture system accessories, the main problem was to adjust the culture substrate concentration for optimal cell attachment to the Prodigy chamber, which took a few trials to get it right. The other smaller issue was scaling up from our usual 6-well plate scale to much larger surface of cell stacks and adjusting seeding densities.
Okay, many thanks, Rafal, for the answer. The next question is, will the CliniMACS Prodigy affect the pluripotency properties of PSCs during the automated expansion process?
Well, maybe I think I can take this question. So because I think I already very briefly showed some of our results in my presentation. Well, it is indeed a very important question. So as I showed in the presentation, so the PSCs that are expanded in the CliniMACS Prodigy adhesion cell culture process, well, they show very high. They show the expression levels of the key pluripotency markers that are very comparable to the cells that are expanded manually. And second, so the trilinear differentiation potentials of the PSCs that are expanded on the CliniMACS Prodigy are also comparable to the manual control. So that's why we believe the quality of the PSCs that are expanded on the Prodigy, on the CliniMACS Prodigy, they are, the qualities are very good.
Okay, thanks, Chao. And then we have the third question I can see. It is, can I use the CliniMACS Prodigy system to differentiate PSCs into other somatic cell types?
Well, I think I showed some results from the MDA and your progenitor differentiation. Well, differentiation into other cell types, may require different medium, small molecule cytokines, but as long as the differentiation process is adherent, and the handlings in the CliniMACS Prodigy adherent cell culture process might be similar. Thus, it's also likely that this can also be adapted and realized on the CliniMACS Prodigy. But of course, then we would need to discuss more details and to see if and how to automate these required processes on the CliniMACS Prodigy. So that's why I mentioned if you are interested in automating your process in the closed CliniMACS Prodigy platform. So just please get in contact with us, and we would be happy to discuss further details with you and your team in a dedicated meeting.
Many thanks. So it looks like that we have time for one more question in today's webinar. And the last question is how to connect the cell stacks to the CliniMACS Prodigy and how to make sure the connection is sterile.
I think I take this question. To connect Corning cell stack to the CliniMACS Prodigy instrument, we need some additional accessories. 33 mm filling cup with lure connector from Corning and 1 meter tube extension from Miltenyi Biotec, which also has lure connectors. We connect both accessories to the cell stack inside the biosafety cabinet, and then the assembly is ready for connecting with Prodigy tubing set by tubing welder. We are using Terumo TSCD2 that assures sterile connection of both wet and dry tubing under the normal condition.
Okay, thanks a lot, Rafal and Chao. So do you have any final comments for our audience today?
Yeah, I think, sorry.
Please go ahead.
If anyone would like to have some talk or send email exchange around the closed culture system or other CiRA Foundation related issue, please feel free to email me or find the information on the CiRA Foundation website.
Yeah, from my side, I just once again I want to thank Dr. Rafal Krol for joining us today and presenting his most recent data on human pluripotent stem cell banking. So if you have any questions, please do not be hesitant to contact us. And we also hope that our presentation today could be helpful for your projects. Thanks a lot.
Okay, thank you again, Rafal and Chao, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. And before we go, I would like to thank the audience for joining us today and for the interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand, so LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So thanks a lot also from my side, and we see us next time. Goodbye.